Gen Bio. Today we are touching base for our first Biology 112 lab. So for spring semester 2022. And unfortunately, we are virtual again. So I have chosen to do this asynchronously. I know some of the other sections are doing it synchronously, but for the sake of anyone who may have connection difficulties or other commitments at home, I've chosen to record this and post it for you guys and allow you to work through it to view it on your own time. Now, I do have to monitor attendance though. I have to track who's attending class. And even though we're virtual, it's a requirement that you virtually attend. So I have decided to ask a number of questions throughout the course of this PowerPoint. And you will pay attention to the PowerPoint. And when I tell you at various points that this is a question, you will write down the answer on a piece of scrap paper that you have handy. And when the PowerPoint is complete, you will know what to do. I will tell you to upload your answers to Canvas. And it'll just be very simple, short answers so that I know that you have in fact watched the video. And that's really the entire point of this. I want to make sure that you are attending class, despite the fact that we're not together in person and we're not synchronous. So just wanted to give you a quick little bit of information about that before we get into the details that we would normally cover if we were in person together in lab. So for most of you, I think I've met many of you. If I haven't, my name's Colleen Fry. I've been teaching this class for, like 11 years now. So I hope that everything will go smoothly. I suspect that it will. At any point, if you have any questions throughout the semester, you can always shoot me an email at cfry@lvc.edu. I do try to respond to those within 24 hours on weekdays and by the next business day if it's a weekend. But that's the best way to get in touch with me. I'll also have office hours once we're back in person. I'll usually be available before or after lab, and we can schedule appointments as needed, and those can be in person or via Zoom, whatever's best for you guys. All right, so for the lab logistics, we're obviously going to use Canvas heavily. I usually do use it even when we're in person for submitting assignments. All of your assignments will be submitted on Canvas. This way I can't lose them. You can submit them up until the due date. So it's most convenient to keep everything in one place and see your grades and be able to go back and look through what you needed to correct. Neither one of us can misplace the assignment that way. So we do use Canvas heavily, so I expect you to be on there regularly. I will also post my PowerPoints, so any of the lectures that I give before we actually get down to the hands-on work for lab, any of those lectures I will also put on Canvas for you guys to have access to. This semester, you may have noticed in the syllabus, there are no quizzes. We had all those quizzes last semester. We're not doing that this semester. We are having writing assignments, however. So if you look through the syllabus, and I can sort of show you that here as well. I think I'm sharing this with you guys. If you look through the syllabus, you'll find my contact information and the textbook that you need, as well as all the assignments. And you'll notice that, like I said, the assignments are writing assignments. There's a plagiarism assignment and a citation assignment to learn how to properly cite your references. And then different parts of a scientific paper. So the first part is the introduction, second part is the methods, the results, and the discussion. And as we work through the semester then, you're going to have practice writing each of the sections of a research paper for science writing. And then towards the end of the semester, you'll put all of those parts together you'll put what you learned in writing each of those parts together into one big lab report that is contains all of those parts and will be on one of our labs that we do towards the second half of the semester. So like I said, these are sort of to take the place of all the quizzes. I do think that in the fall, there's an advantage to the quizzes in that it teaches freshmen, which most of you are, that you need to stay on top of the material. Okay. And hopefully by now you know that. So you can stay on top of the material and study weekly, keep a pace, don't let it all go till the exam. But hopefully you know that by now. So we're not going to be doing the quizzes. And like I said, we're going to be stressing science writing this semester instead. So those will be a major part of your grade. There's, like I said, the 4 different parts of the paper that are worth 10 points each. And then at the end of the semester, you'll have a large writing assignment, which will include all those parts on a different topic. So that'll be 50 points. And then you have 3 lab exams this semester. So they'll be 50, 60, and 40 points respectively, and they'll cover the labs listed here. You can find all this information in the syllabus as well. So same as last semester, we're going to combine your grade for lab with your grade for lecture, and that will give you one whole grade for both. So that's the basic format for how we're going to have lab. Like I said, you should be on Canvas regularly. You should be attending. If we have some situation where you cannot attend due to illness, or I know some of you guys are on sports teams that may have to travel for meets, then what you should do is go to the last page of the syllabus, where you can find, if I scroll all the way down, where you can find all of the instructors and their email addresses. So all of these different sections of the lab are taking place throughout the week. And luckily we're on Tuesday, so you can find another time, Wednesday or Thursday, when you're able to come to lab that week. Or if you know ahead of time, you can also choose a Monday lab, and contact me as well as the professor whose lab you would like to attend. So you would have to reach out to both of us. We are under some constraints as far as how many students are allowed in the lab at a time. So you need to contact the other professor to make sure that they have an open seat that you would be able to utilize for that week. So like I said, that's basically the logistics. If you have any problems with any of that, you can always shoot me an email, or any questions, shoot me an email, and I'll be happy to answer them. All right, so let's get into the nitty gritty then. For week 1, we are looking at protostomes. And if we were in lab in person, we would be dissecting the crayfish and the Nereis as model organisms from the group called protostomes. And before I get into exactly what a protostome is and get into the details about crayfish and Nereis, I want to make sure that you are all familiar with directional terms. So when we look at specimens, when we dissect specimens in the lab, you need to know sometimes which direction I'm referring to. If I say that, oh, the heart is caudal to the stomach, you need to understand what I mean by that. So these terms are important for you to recognize and know if you don't already. So I'll just talk you through them. Rostral means towards the nose. Caudal means towards the tail. And that can be on a 2-legged organism or a 4-legged organism. Either way, rostral is towards the nose, caudal is towards the tail. And then dorsal is towards the back, okay? And again, this is on a 2-legged person, 2-legged animal that's sort of upright in the air, right? Vertical. It would still be towards the back on a 4-legged organism, but it would be in a horizontal plane, right? Towards the sky, right? So dorsal is towards the back, ventral is its opposite, towards the belly. And then median means midline. It would be just like down the center of the organism. And lateral means to either side. If I say it's lateral to that, it could be to the left, could be to the right, but it's away from the midline. Okay. And then sometimes I might use terms like proximal, which means closer to the body, and distal, which means further from the body. So this is my first question then that is going to be used to gauge attendance. So when you so write this down. The first question is, what term refers to towards the nose, right? And your answer you should be able to answer already, is rostral, right? So write that down. When you submit your answers on Canvas, you can just write the word rostral. Number one, rostral. Okay, and I'll know that you attended lab. Now, that's not the only question I'm going to ask. I'll tell you two more throughout the course of the lecture today. All right, so let's move on then and look at a phylogenetic tree. This is basically a tree that compares how related different organisms are, right? And so for this phylogenetic tree, we're looking specifically at animals, and not all of them, but a large number of categories are present on this phylogenetic tree. There's actually 4 different branch points that we're going to look at, which will lead us ultimately to the protostome group and the deuterostome group. This week, we're looking specifically at protostomes. And then in week 2, lab 2, we're going to look at deuterostomes. So we will look at those, but today we're focusing on members of the protostome group. But you should know a few identifying characteristics along the way on this tree. So there's different branch points, right, which indicate that there's some divergence, some separation from a common ancestor. So at the very beginning of our tree, we have what we believe is the ancestor to all animals in this group. So we have that ancestor, and then there's a divergence point where we see some animals that are called parazoa, or not really true animals. Para means, closely related or sort of, right? Parazoa means they don't really have true tissues, versus the eumetazoa, which do have true tissues. So again, tissues are groups of cells that work together for a common function. You might have nervous tissue, right? Or muscular tissue, right? So all of those tissues are more distinctly present in the eumetazoa group. And the parazoa, they include some sponges that don't really have real tissues that work together for a specific purpose. All right, and so if we then follow our eumetazoa group, true animals, we can then see the second point of divergence, which is referring to symmetry and germ layers. So symmetry is a easy to understand concept, right? If the organism has radial symmetry, that means it can be cut on any plane, right? In a circle, basically, that will show equal halves, right? You can cut it in half on any plane, top to bottom, and you'll see that it has symmetry. Versus bilateral symmetry, which is the second group here, means that you can only cut it along one plane. Usually there's a left and a right order of symmetry, where if you could fold it in half like a piece of paper long ways and see that the left and right are identical, but it would not be identical if you cut it, say, if you cut a human in a horizontal plane, we obviously are not symmetric top to bottom, right? Okay, so jellyfish are an example of the radially symmetric animals, where you can cut them basically on any plane in that top to bottom fashion, and they'll be symmetric. Of course, humans are an example. Also, all of the organisms that we're going to look at in the the deuterostomes and the protostomes today, as well as next week, they are all members of the bilateral symmetry subgroup. Okay, so we're following this sort of branch on the phylogenetic tree. As we progress then, we're going to see a divergence in whether or not the animals have body cavities. These are also called coeloms, C-O-E-L-O-M, the presence or absence of this body cavity. And this cavity is something that's within the body wall, but outside of the digestive tract, right? So we have a tube, that's our digestive system, mouth to anus, that runs down the center of our body. And then around that, there's a space. Now we have other things in that space sometimes, but it should be a space that could be fluid or air filled, that is within the body wall. but outside of the wall of the digestive tract. So that's what a coelom is. And so some organisms have it, some don't. So there's a distinction here in our tree. Acoelomates do not have a body cavity. Pseudocoelomates have a body cavity that is not enclosed by mesoderm, which is one of our germ layers that we had learned about last semester. And then coelomates are those animals that do have body cavities that are completely enclosed by mesodermal tissue. So that germ layer, all of the organisms that we're going to look at have that mesodermal tissue. So they are triploblastic, we call them, 3 different germ layers. Whereas some other animals only have 2 germ layers. They would be diploblastic. They would just have that endoderm and the ectoderm, versus triploblastic animals that also have a mesoderm. All right, and so that brings us to the last point of divergence, right here, where we see then the distinction between protostomes and deuterostomes. And this distinction is based on a number of factors. The first one is how that mesoderm tissue forms. In protostomes, which are the ones we're going to look at today, the mesoderm, it kind of splits off from, and we'll look at this on the next slide, some little cell masses near the blastopore. Okay, we'll look at this in a picture in the next slide. And then in deuterostomes, then the mesoderm forms from outpocketings in the archenteron, which, if you recall back from last semester, that archenteron was the continuation of the blastopore. We had looked at this in slides of C-star development. So anything that is grouped with the protostomes has that mesoderm tissue forming from little cell masses that are around the blastopore. And also for protostomes, another important distinction is that the mouth forms from the blastopore. So proto means first, stoma means mouth. So the first opening into that ball of cells during embryonic development is that blastopore. And in protostomes, that first opening becomes the mouth. In deuterostomes, deutero means second, and stoma, again, means mouth. So in deuterostomes, then that first indent, that blastopore, becomes the anus, and then the second opening is what will become the mouth. Okay, so there are 2 big distinctions then in the development of protostomes and deuterostomes. So we classify them according to those 2 differences. All right, so this is showing you in a nice picture here kind of what I just said. You can see that in both protostomes, which we're going to talk about today, and deuterostomes, which we're going to talk about next week, there is the presence of a body cavity within the body wall. Okay, so this blue is the outside, will become the body wall for both protostomes and deuterostomes. And then there's a digestive tract that will ultimately form, right? The yellow is going to become their digestive system. And then the mesoderm is in between. Okay, that's going to be the coelom. Okay, and that space in there, like I said, that's the coelom, forms from the mesoderm cells, which for protostomes, again, sort of form these masses that originate near the blastopore. Okay, and then for deuterostomes, the mesoderm buds off from the archenteron. If you remember, the blastopore leads to the archenteron, and at the sort of the terminal end of that, there's a couple of cell groups that sort of bud off and start to migrate, and then line this space, the coelom. Okay, and so those will be deuterostomes, and the others will be protostomes. Okay, and like I said, the other big difference then between protostomes and deuterostomes is that blastopore, that first indent into the blastula, will become the blastopore will become the mouth for protostomes, and the blastopore will become the anus for deuterostomes. So another developmental difference. Now, today we're just looking at protostomes, but you should be aware of the differences. So today we're looking at those organisms that have that blastopore forming into the mouth. So again, proto is first, stoma means mouth, first mouth. And the two model organisms that we have for you in lab today, well, in 2 weeks, are the Nereus Worm and the Crayfish. These are both protostomes. They both have bilateral symmetry. So if you cut them, you can only sort of cut them one direction, right? From the head to the tail, and they'll be symmetric left to right. They also have another feature called segmentation, where they have these different repeating body segments, right? And we call those metameres. And most of the body segments, most of these metameres have appendages. This is true for both the Nereus and the Crayfish. You'll see a lot of appendages coming off. There's a heck of a lot more specialization in the appendages and in the development in the Crayfish than there is in the Nereus. The Nereus worm has the same appendage pretty much, except for a little bit towards the head, the same appendage on every single body segment. So it's just kind of repeating segments. Whereas there's some specialized function that is then that has then developed in the Crayfish from the Nereus. Okay, so we'll look at the Nereus first because it's the more simplistic. I think your lab manual actually does it the other way around, but we will look at the Nereus first. It is a member of the group Annelida, which annelid means ring. So that's because of these repeating segments, these repeating rings that make up the segmented worms. And it's part of the class Polychaete, which means many bristles. So I've also heard it said polysetae, because those bristles are pronounced setae, the little bristles that are coming out of the appendages or the Peripodia on our Nereus worm. And its genus is, of course, Nereus. So like I said, it is segmented. It has these repeating rings, these repeating metameres that go along the entire length of the body. So let's look at it in a little more detail. Another neat feature, if we're just looking at the external anatomy of the Nereus, is that the Nereus has the ability to evert its pharynx for feeding purposes, meaning that if you are looking at a worm that is not feeding or was not killed while it did not die while feeding, it would look a lot like this. The head is here. You can see that there are these repeating body segments with little appendages coming off of each segment. The appendages are called Peripodia. But if you look at the head region, you'll see that there are some specialized appendages here, which we'll talk about, but that it does not have its pharynx very visible. The pharynx is basically a continuation of the mouth, right, where food will travel to go down through the esophagus and then the intestine for processing of the food. But in order to eat, the Nereus is able to extend its pharynx outside of its mouth. So it kind of just like extends it out, is able to grab food and pull it back in, right? And if it were killed while eating, if we were able to obtain a specimen with the everted pharynx, it would look a lot like this top picture here. We can see these are the head appendages, which are present here in the Nereus with the inverted pharynx. They kind of flip up, and then the pharynx itself is stuck out through the mouth, right? And on that pharynx, then you can see these jaws, which are these little graspers, little pinchers, which allow the Nereus to grasp its food and pull it back in. There's also some little black dots you can kind of see here. Make it a little bit bigger, maybe you can see a little bit better. These little tiny black dots, if you actually look at the Nereus, you can feel those. They're little bumps, or basically like little teeth that help to grind the food up. They're called denticles. Okay, so you want to look for those when we actually have the specimens on hand. And so that's a neat feature if we're just looking at the external anatomy. We're going to look at a little bit more external anatomy in our next slide. I talked to you about the fact that there were some specialized appendages in the head region. Most of the appendages are called Peripodia, okay? And they are on almost all of the body segments. If you look though towards the head, there's a segment that is sort of just in front of the first segment with Peripodia. Okay, so there's a segment that's just in front of that, and that is called the peristome. That is just a junction basically between the head and the rest of the body. Okay, so the peristomium. And then just in front of that is the prostomium. And you can also see, you can see the peristomium right here. The prostomium has these other little sort of bilateral nodules on it called palps. It also has eyes, little black dots here. And then several specialized appendages coming off of the head region. Okay, and these are called tentacles. They are used for feeling the environment and recognizing food sources. So those are specialized areas in the head. If the pharynx were everted, as I had mentioned, you would also be able to see the jaws. But if the pharynx is not everted, rather inverted, then you would not be able to see the jaws or the denticles. They're shown here, but that's again showing you the everted pharynx, which you can see here as well. So these nice little pincher-like structures that help it grasp its food and bring it down into its body. Okay. All right, and then after we examine the external anatomy, which again is just, we're looking at the head parts. Hopefully we'll be able to have a specimen for you with the everted pharynx, and then along the length of the body, all of the metameres with Peripodia. That's really as detailed as we get for the external anatomy. We will then do a little bit of dissection for the Nereus, where we're going to cut basically down the length of the body, cut it from left to right, and then cut up so that we can separate out the intestine and see the intestine, separate it away from the body wall, which is right here. And we're going to pin back the body wall. Okay, and you'll be able to see then some internal anatomy. For most of your specimens, it will have an inverted pharynx. So it will look something like this, where you're going to cut up the length of the body and pin back the body wall. And then you should see the pharynx, which has a sort of a roundish appearance to it. And then just behind that is the esophagus, which is a little bit more narrowed than the pharynx. It kind of elongates and narrows a little bit. On either side, you'll see what we call esophageal cecae is plural, or cecum is singular. So there's one little, it's kind of hard to see here. Let me make it a little bit bigger. Maybe you can see it a little bit better. A little gland here, basically, that secretes digestive enzymes that help the Nereus start to digest its food. Okay, and then if you follow the esophagus back, you'll see the intestine. So the rest of the body, if you go down the whole rest of the body, the intestine will travel through there until it gets to the anus. And then just on top of the intestine is the dorsal blood vessel. If you pull up the intestine and look underneath it, you'll see the ventral blood vessel as well. I should mention that we cut it on the dorsal side, and so you would see the dorsal blood vessel. So you would put it with its mouth parts down on your dissecting tray, and then the first blood vessel you would see would be the dorsal blood vessel. If you pull up the intestine, then you would see the ventral blood vessel underneath that. Okay, so those are the main structures that you will see in our Nereus dissection. There's one more I can think of at the moment as well. So if you look, again, you're pulling up the intestine, you'll see that ventral blood vessel. You'll also be able to see a ventral nerve cord. I think we talked about a little bit last semester that some organisms have dorsal nerve cords, such as ourselves. Our nerve cord or spinal cord is on the dorsal side, whereas for protostomes, that nerve cord is still ventrally located. So if you are looking for its nervous system, you're going to have to look underneath the intestine on the ventral side. Okay, so it'll be running sort of this side of the body towards the chest or the abdomen. And so you will look for that as well. Now, we can distinguish between the ventral blood vessel and the ventral nerve cord based on color. The ventral nerve cord is a lighter, paler color versus the blood vessel is usually a little bit darker. I'll direct you to this next slide, which is basically a picture of a microscope slide that you are to examine this week. Now, since you're not in lab, we have made videos for you. I've made videos that kind of talk you through looking at the two slides that you would have examined if you were in lab today. So this is one of those slides, is a picture that looks very similar to one of the slides. It is a cross section through the Nereus. So if you were to take your Nereus and cut it this way, sort of left to right, cross section, and then turn it and look at the cut section, right? Look at the surface that you just cut. It would look something like this, where you would see this is the dorsal side and this is the ventral side. You would see that there's, first of all, an intestine in the very middle, right? J is our intestine. There's a body wall, of course, on the outside. There are Peripodia, those little appendages that come off on both sides, left and right. And then there's some other structures, some muscles and blood vessels and nervous system within the body wall. Okay, so we're going to identify each of these different structures. First of all, let's talk about muscles. So there are two sets of muscles that run down the entire length of the body, like head to tail. And those are the dorsal and the ventral longitudinal muscles. Okay, so of course the dorsal one is going to be on the dorsal side, the ventral one is going to be on the ventral side. So dorsal longitudinal muscle, and again, they're both present bilaterally, left and right, is letter A. And then the ventral longitudinal muscle here and here, both bilaterally, that's letter D. Okay, so that'll again run the whole length of the body on the ventral side. And then you also have two other muscle groups. They're not running longitudinally though. H here is our oblique muscle. Okay, so you got that branch sort of starting in the middle along the ventral midline and extending to the sides, both sides, the oblique muscles. And then also there's a circular muscle, which is, you can sort of see it right here. The circular muscle goes around the entire outside of the organism, but inside the body wall, but sort of outside of the longitudinal muscles. And it helps to allow for movement by enabling a constriction or sort of a dilation of the entire body of the worm. Okay, so it is circular in nature. Okay, so those are the muscle groups that you should be familiar with. I had mentioned the intestine, which of course is used for processing food, is J. Just outside of that is the space, the coelom, that is in between the digestive tract and the body wall. So that's letter G here. And then we have a couple of blood vessels that I had mentioned on our last slide. I is the dorsal blood vessel, and F is the ventral blood vessel. Okay. And then just underneath the ventral blood vessel, like I said, that's where you're going to find your ventral nerve cord, letter E. Okay. All right, so that's basically the internal anatomy of the Nereus worm. You should look at the slide video that is also posted on Canvas for you, because it'll help you to observe the fact that you might not be able to see the entire cross section in one field of view when you're looking through the microscope. So if we give you a portion, you might only see, say, the upper right corner of it. You should still be able to identify the parts that are visible in that field of view. Okay. And then the second slide, microscope slide that I've recorded for you and posted on Canvas is a microscope slide of the Nereus Peripodia. The Peripodia, I've mentioned a couple times now, are these appendages which are on each of the metameters, each of the body segments of the Nereus. And they have a role in locomotion. They help They help the Nereis move, but they also provide an increased surface area for the worm, and this serves to help with respiration for it because they don't have any proper gills or lungs that help it to breathe. They use this increased surface area of the Peripodia as just a respiratory surface diffusion across the membrane. So this is a slide that shows you a detached parapodia. It would be connected to the body wall right here. And there's sort of two parts to it. The top and the bottom, or dorsal and ventral parts. They have specialized names, okay? So the fact that there are two parts, the fancy name for that is the fact that it's biramous. So two parts, biramous, two branches. The top is the Neuropodium, the more dorsal side. And it is usually larger. And it, as well as the ventral portion, have sort of two parts to it. So this and this these are both part of the Neuropodium. The more ventral section, again with two parts to it, is the Neuropodium. And you can remember this by the fact that the Neuropodium is closer to the nervous system, right? The Neuropodium is referring to the fact that it's closer to the nerve cord, the ventral nerve cord. So I'll help you remember which one's which. Again, there's two parts, an upper and a lower ligule to both the nodo and the Neuropodium. And then there are these chitinous support rods, these structures that just help with support of both the nodo and the Neuropodium. Those are called Akikula. Okay, so these little dark stained branches right here, you can see those are two Akikulae, and they function as support. And then you'll also see these bristles right here. These are called setae, and each of these bristles can help with movement again and sensory functions. They each arise from a single cell and form these branches that then help the organism move and sense its environment. Okay, so the main structures you have to know for the Peripodia is just the nodo and Neuropodium, the Akikulae, and the setae. Okay, I just tell you that there's two parts to it, just so you don't confuse the bottom part of the Nodopodium as the Neuropodium. Okay, so that was all part of the Nodopodium. All right, so that wraps up the Nereis. We are now going to move on to the crayfish. And the crayfish is also a protostome, right? So it's somewhat related to the Nereis, and you'll see some similarities in the fact that there are these repeating body segments. There are appendages on most of the body segments for the crayfish as well. However, of course, there's some increased specialization. So a little bit about the crayfish. It's an arthropod, which means jointed foot. You'll notice that each of the feet have different segments to them. So joints between the different segments. It's in the order Decapod, which means 10-footed. It has this very noticeable exoskeleton, this hard outer covering that provides protection for the crayfish. It lives in an aquatic environment and needs to be protected from predators in that environment. And so the exoskeleton serves that purpose. As the crayfish grows, it will undergo several stages of molting where it needs to shed its exoskeleton and grow a new one, basically. And so during that time, the crayfish can be somewhat vulnerable to predators because it doesn't have as strong of a support structure, as strong of a protective outer layer that keeps it from being an easy target for predators. On the dorsal side of the crayfish, the covering this exoskeleton is called the carapace. Okay, so on this dorsal section, we call it the carapace, and then there's a few large sort of gross divisions in the anatomy for the crayfish. The head is one, the thorax is another, and they are divided by what we call a cervical groove. This little line here is just a groove, but they're actually joined. It's not a separation because these two sections of the exoskeleton are actually attached. So we call the head and the thorax together, we call that the cephalothorax. And then there is a physical distinction between the cephalothorax and then the tail region, which we properly call the abdomen. Okay, so the large anatomy, if you're just looking at basic structures, you've got the cephalothorax, you've got the abdomen, the exoskeleton is the protective covering. On the dorsal side, that's called the carapace. And then we're going to start looking at body segments. So a little bit more specialization is going to come into play here. There's obviously a lot more specialization around the head, and with the appendages, you're going to see a lot of different characteristics to each of those. Like I had mentioned, most of the body segments have an appendage attached to them. So the crayfish has 19 body segments that we call metameres, and you're going to find appendages on most of those. There are 13 sets of appendages on the cephalothorax, okay? So when I say that, I mean that there's a left and a right, and so that makes up one pair. There are 13 pairs on the cephalothorax, and then 6 pairs on the abdomen, and we'll look at each of those. So let's move on to our next slide. That was the dorsal side, right? So now we're going to flip it over, basically, and look at the ventral side, or the underside of our crayfish. So for when we first look at the crayfish, the first thing that you want to look at on the ventral side is you want to identify if you have a male or a female. There's a difference in the appendages that will help you tell the difference right away. Then there's also some differences in the reproductive anatomy that you'll be able to observe as well. So if you're looking towards the head first, you'll see that there are these antennae and antennules. There are some eyes and then some mouth parts. The mouth parts each have appendages as well, which we're going to look at in more detail in just a minute. But then you'll see these big, large, what we call calipeds, these big grasping appendages, help it to grasp onto food and start to crush it. So those are called calipeds. And then you'll see 4 sets of walking legs. Let me zoom in on this a little bit for you. There are 4 sets of walking legs, 1, 2, 3, and 4. And these help with locomotion for the crayfish. And then there are 5 sets of what we call swimmerettes or pleopods. They are on the abdomen, the underside of the abdomen. And this is where you can start to see the difference then between the male crayfish and the female crayfish. I'll show you that in just a sec. But I'll just mention that then you also have appendages on the very end of the tail, the uropod and the telson. And this picture here will show you all of those in more detail. So the uropod is present bilaterally. The telson is the very middle portion of the terminal end of the tail there. And then 1, 2, 3, 4, and you can't really see, but there's a 5th parapleopods or swimmerettes that's present on the abdomen. And this is, like I said, this is how you can tell the difference between a male and the female, because the male has, and I'll just flip to the next slide to show you this, the first 2 pair of swimmerettes or pleopods much larger than the rest. So you can see this is a little bit blown up. The picture's blown up. You see these 2 here are the first and second. I don't know if you can see here, but this is one, and then just underneath that there's another first and second pairs of swimmerettes or pleopods. So the male has those being larger, and then the last 3 pair being smaller. Whereas the female I'd started to point out here, you see has 1, 2, 3, 4 that are roughly the same size, 4 pair, and then one tiny little pair up here. You can barely see it, but it's there. So the female, the first sort of the most rostral pair of swimmerettes is small, and the latter 4 are large. Whereas in the male, the first 2 pair here and here, 1 and 2, the first 2 pair are larger, and then 3, 4, and 5 are smaller. 1, 2, 3 are smaller in the male. So if you look at your crayfish, when we actually are able to do these dissections in lab, you'll see that there's a size difference in the pleopods or the swimmerettes between the male and the female. So you should be able to recognize just from that whether or not you have a male crayfish or a female crayfish. And then before I move on to the next slide and show you those in a little more detail, I'll mention that in the video that Jen Thornsberry posted on Canvas for you guys, she has kindly recorded herself doing dissections of both the crayfish and the Nereis. But in the video of the crayfish, I think she told us that she thinks she accidentally called the Europod the Telson and the Telson the Europod. So my second question for you to track attendance is for you to answer which appendage on the crayfish is not present bilaterally, okay? And so the answer is Telson. The Telson is the center one. It actually holds the anus. This is where, of course, all the waste, the food waste, will exit the body. And so that Telson is the center one. It's not present bilaterally, but the Europods are. Okay, so there's a left and a right Europod. She said she thinks she messed that up in the video. So that's my second point. I want to make sure I tell you that it is correct in the lab manual. So that's right. The video might be wrong. So again, my second question for attendance tracking is which tail appendage on the crayfish is not present bilaterally. Okay, and so the answer is Telson. All right, so let's move on to our next slide then, where I'd kind of shown you already. This is one of the 2 larger pair of swimmerettes you can see in the males. The latter 3 pair of swimmerettes are smaller in the male. Okay, and so these first 2 pairs are called copulatory swimmerettes. They're used in fertilization in order to help the male fertilize the female. So they're called copulatory swimmerettes. And then if you look at the female, you can sort of see here that this is a tiny little pair, I believe, right here. You can see it's kind of trailing off to the left. That's one of them. And then the second, third, fourth, and fifth would all be larger swimmerettes than the first pair. Okay, if you compare them to each other. All right, and then also in females, you're going to see a couple of openings. Number 2 in the picture here is the opening to the seminal receptacle. This allows for internal fertilization, which is characteristic of crayfish. And then number 3 here is the opening to the oviducts, where the eggs are ultimately released from after fertilization. So you can see that they're present here. This is a very idealistic picture. In your specimens, they will not be nearly so noticeable. They've been expanded just to make them more visible, and you know that they're there. But if you look at your specimen, usually around the location of the last walking leg, you're going and be sort of around the first pleopod or swimmerette, you'll see then that opening to the seminal receptacle. You'll be able to see that. And if you sort of palpate with your probe, you might also be able to see the openings, the oviducts at the base of the walking legs. So you can look for those. Again, this is an idealistic picture, so don't expect to look that great on your specimen, okay? You got to kind of hunt for them. But you should be able to see them and find them. And then this is just showing you that once the eggs are released, they actually attach to the swimmerettes until they hatch. And this is just showing you some of the the eggs that are still developing, ready to be hatched after development, and then they will be released into the environment. Okay, so that's the anatomy as far as appendages on the large scale, as far as the walking legs, the calipeds, and the swimmerettes. Now we're going to look at some appendages that are associated with the head and the mouth. So there are 6 pairs of mouth appendages, and then 2 other pair of appendages on the head. The most rostral are the antennules and the antennas. The antennas are the larger ones, and the antennules are smaller. And then the other ones shown here are very, very small, and they cover the mouth. So the mouth, again, we're looking at the ventral side of the crayfish. The mouth is here, and it's covered by all of these pairs of mouth parts, mouth appendages. So when you have your crayfish in lab in a couple weeks, you're going to take very carefully, you're going to take your tweezers and you're going to remove one side of the mouth appendages. You're going to work from the sort of the more caudally attached mouth parts towards the more rostrally attached mouth parts, because they kind of like lay on top of each other. So you got to work from the back basically. So you're going to take off the third maxilliped first. It's going to be the outermost one. It's the largest. This is probably it right here in our picture. So you're going to take that off first. You're going to kind of grasp it with your tweezers and just pull on one side only. So you leave the other side intact. And then once you get that off, you're going to then look for the next appendage, which is the second maxilliped. Just underneath that is the first maxilliped, then the second maxilla, the first maxilla, and the mandible. The mandible is a very hard, if you tap it with your tweezers when you're taking it apart, it's a very hard structure. It sits right on top of the mouth. All of the others are a little softer, and the maxilla, the first and second maxilla in particular, are very, very soft, very thin, and very feathery like. So when you're detaching those, you kind of have to really tease out which appendage you're actually looking at. If you get down to the very base and you look for the fact that there's a separation first before you remove it, you should be able to identify each of the different mouth parts. But it does require a little bit of care and time to tease out which belong to which mouth parts, because again, these are biramous also, so they have 2 parts to them. So you want to make sure you get both parts that belong to the same mouth appendage, and not sort of rip off half of it and confuse it with the next one. Okay, so we're going to have you take each of those off in that order, working from the third maxilliped, which is the outermost, most caudally attached one, working from that to the mandible, okay, which is just on top of the mouth, and it'll be harder. So you'll definitely be able to recognize that one. So you're going to remove the appendages on one side, and then start to look at the internal anatomy of the crayfish. So if you cut then the carapace off of the cephalothorax and you just remove that, this is basically what you will see then. You'll see a lot of structures in here, many of which are associated with the digestive system. Okay, so just if you take you might take a pair of scissors and cut. You can cut along the midline or along the side and sort of remove that carapace. You're gonna remove it also by cutting from the ventral side up towards the dorsal side, and then down and removing. You only have to do like one side, the left or the right, if you want to leave the other side intact for structural support. That can be helpful. But if you want to remove a little bit more so you can see the entire internal anatomy, that might be helpful as well. So if you remove that carapace, like I said, there's going to be a lot of room taken up by the digestive glands. They're sort of like a pale yellowy, pale yellowy, off-white color to them. And then just rostral to that, you'll see the stomach. And the stomach is actually made up of 2 different parts, the cardiac stomach and what we call the pyloric stomach. The cardiac stomach is more rostral, so it's sort of towards the nose, The pyloric stomach is more caudal, more towards the tail. And then if you actually cut open the stomach, then, especially in the cardiac stomach, you can make a little incision in it. You'll be able to open it up and see the gastric mill, which is little teeth, basically, little hard things, little teeth that are inside there that help with mechanical digestion in the crayfish. So helping to grind up its food. And then as food passes from the cardiac to the pyloric stomach, this is where we start to see some chemical digestion of the food taking place. All right, and then you would be able to follow that stomach down and see the intestine, which would then run the length of the body. It sort of transfers from being on the dorsal side to the ventral side and exits through the anus in the tail. So that, and then also if you remove the stomachs and look more towards the mouth, you can see the esophagus, which is the tube that connects the mouth to the stomach. Okay, so that's the digestive system. You should be able to visualize the entire thing from mouth to anus. And then the digestive glands, you'll want to remove those. Just behind or just caudal to the digestive glands is the heart. The heart is the pumping organ of the crayfish. In contrast to the Nereis, which doesn't really have a heart, the crayfish does have a heart. It pumps fluid throughout the body, but I should mention that the circulatory systems are different for the crayfish and the Nereis in a couple different ways. First of all, the presence of a heart in the crayfish and not in the Nereis, and the fact that the crayfish has what we call an open circulatory system versus the Nereis, which has what we call a closed circulatory system. A closed circulatory system means that all of the fluid, all of the blood, is enclosed in blood vessels, and that's what the Nereis has. The crayfish, on the other hand, it has a heart and it has some blood vessels, but there is only a sort of a partial enclosure to that fluid. The fluid for the crayfish we call the hemolymph. It is pumped through some blood vessels, and then it is just responsible for washing over the organs and returning to the heart through little pores in the heart, little openings called ostia. Okay, so if you look at the heart, you'll be able to see, hopefully, that there are little openings. Those are the ostia. And you also see that it does have some blood vessels, but the fluid is not completely enclosed in those blood vessels. So we call that an open circulatory system. Okay, so you'll want to look for that. If you then have identified all of the digestive system, you've identified the heart, you can remove some muscles and a lot of the gills, which are shown here. When you remove the appendages, a lot of the gills will come out with them, but if you have to remove a few more gills, you can remove those. And then what you'll see is the ventral nerve cord that runs along, again, the ventral side of the body. It's going to be a pale white cream colored strand that runs the length of the body. And then you'll also, towards the head region, be able to see what's called the green gland. It's not actually green. It's, again, more like a cream color. Small little gland that is just sort of behind the eyes. Okay, and this is responsible for fluid waste, the excretory system of the crayfish. So it produces fluid waste, which then exits right by the green gland, right out to the outside of the body, which, again, the green gland is here, and then it will excrete its waste, kind of gross, right around its mouth area. So gross. But let's move to our next slide, where you can see some of the structures that I just talked about. So again, if you remove the digestive system, you remove the heart, you remove some muscles, you'll be able to see the ventral nerve cord, this fiber that runs the entire length of the body. It does sort of branch and form a ganglion towards the head, which is this compilation of cells, basically, is basically the brain for the crayfish. And then, like I said, the green gland, you can see it here. It's not green. it's present bilaterally, these two structures here, which are responsible for excreting liquid waste from the crayfish. So I think we've talked about all of those structures now. I will tell you then the last question that I want you to answer for attendance tracking this week is, what structure is responsible for excretion of waste in the crayfish? So the answer is the green gland. Okay, so when I put that, I'll put that assignment on Canvas. You just upload a document that has the answer to those 3 questions, and you'll be good for attendance then for this week. All right, so this is a compilation of the function of organs that we talked about in the crayfish. So the heart, of course, pushes that hemolymph that I had said through the blood vessels. There's a dorsal and a ventral artery that you should be able to find. Again, one's on the dorsal side, one's on the ventral side. They supply the dorsal and ventral parts of the body with oxygenated hemolymph. And then the mandibular muscle is a muscle that moves food so that, excuse me, it's a muscle that moves the mandible in order to allow the mandible to crush food. And then the cardiac and pyloric stomachs are where mechanical and chemical digestion occur, respectively. Like I said, the gastric mill is this hard sort of teeth-like structure that grinds food. It's present within the cardiac stomach if you cut open the cardiac stomach. The digestive glands, they help secrete digestive enzymes that help with chemical digestion in the pyloric stomach. And then, of course, the 2 tubes that lead to and from the stomach, allowing food to travel from the mouth to the stomach is the esophagus, and allowing food to travel from the stomach to the anus is the intestine. The intestine also has a role in absorbing nutrients. And then lastly, like I said, the green gland collects and excretes the fluid waste or urine from the crayfish. All right, and so we are coming down the home stretch. The last sort of comparison slide here is showing you different features that are similar or different in the crayfish and the Nereis. So they both have bilateral symmetry. Like I had mentioned, the circulatory system is a closed circulatory system for the Nereis and an open circulatory system for the crayfish. They both have complete digestive systems, mouth to anus. They involve the esophagus, the intestine. You also see a stomach in the crayfish. And then the respiratory system for the crayfish, there are gills, which you will see very obviously when you remove the appendages. A lot of those leg parts will have gills attached, and those will kind of like pull out with the appendage. And then for the Nereis, their Peripodia function as the increased surface area or gills for the Nereis. Reproductively, they each have one gonad. The excretory system for the crayfish, it's the green gland. For the Nereis, there are metanephridia, and these are very hard to visualize in the Nereis dissection. So we often do not see them. We will take a look and see if we see them, but just know that there is this function, the excretory function is served in the Nereis by what we call the metanephridia. The nervous system, they both have a ventral nerve cord and then ganglia, basically compilations of cells that serve as their brains. And then for locomotion, the Nereis has the Peripodia, the appendages on each of the metameres. And then for the crayfish, they have walking legs, the larger sets of legs that are on the cephalothorax, and then the swimmerettes, which are on the abdomen. Okay, so those are what they use for locomotion. All right, that is wrapping things up. So for this week, you will look over, read your lab manual, watch the videos. There are the 2 dissection videos and the 2 slide videos that you should familiarize yourself with. Know the external and internal anatomy for both the crayfish and the Nereis. The Nereis has the 2 slide videos that you should also look at. One's a cross section and one's a slice through the Peripodia. And then for the crayfish, you should be able to tell the difference between the male and female. So watch these videos. You'll be able to see them again there. Jen looks at the external as well as the internal anatomy. You'll be responsible for identifying the appendages. And then lastly, on your radar is that you need to submit the answers to the questions for attendance tracking. And then there's the 2 writing assignments that are posted on Canvas. One is plagiarism commitment that you will not plagiarize. You know what plagiarism is, and you will not exercise this practice. And then the second is a citation writing assignment where you will work through the instructions to learn how to properly cite a scientific journal article or the lab manual or a textbook. So you will look at how to cite those as references when you write your papers. So those are your first assignments for this week. Any questions, you can always shoot me an email at cfry@lvc.edu. And I hope you guys have a great week, and I'll hopefully see you soon.